The biggest story in sequencing this year, next on Mendelspod. Welcome to the program, everyone. I'm Theryl Timpson. Our guest today is Mark Kokoris, the inventor of Roche's new sequencing by expansion, or SBX technology, and the head of the company's SBX program. Roche announced their new technology at AGBT this year, and the write-ups keep getting better. A few weeks ago, the SBX platform made global headlines when the Broad Institute, working with Boston Children's, used it to set a new world record for genome sequencing, from blood sample to variant call in under 4 hours. It's the kind of benchmark that could again shift the world of sequencing. Mark has spent much of his career at the intersection of chemistry, molecular biology, and invention. His vision of creating a next-generation DNA sequencing technology that would enable broader use in healthcare led him to co-invent the SBX technology in 2007. That same year, he co-founded Stratos Genomics, where he held roles as president, chief science officer, and then later as CEO. In 2020, he led Stratos through the company's acquisition by Roche, where the SBX chemistry could be brought together with Genia's massive parallel IC technology. Mark now heads all of SBX research at Roche. He's an inventor of over 20 issued U.S. patents, and now he's leading what many are calling the next major leap in genomics. Welcome, Mark.
Thank you so much, Theryl, for having me. Yeah, it's great.
Our pleasure. Our pleasure. I tried to give you a nice big introduction there.
Yeah, thank you.
So you're a co-inventor of SBX, but I would love to start with a little history. I've never known that much about Stratos, but I knew Genia really well, and we had Stefan Rover on the program multiple times before and after that acquisition. And I've been over to the Pleasanton facility, but then it just kind of it was just kind of a period where we didn't know what was going on. Right? Would a technology emerge from this actually? And it did, and you've delivered big time. So what are some of the highlights for you looking back on what got you to today?
Yeah, it's been quite a journey, I can say. So I like to call it SBX a wildly ambitious chemistry. But I think Keith Robeson had a better way. He said he referred to SBX as a bar bet between chemists, which I thought was.
Oh, okay. So what would be the bet?
Yeah, is this even possible to do? The
Okay.
chemistry was so outlandish. I've shown structures before, and you can literally hear people gasping at what I was showing, because it is kind of a new way of looking at things about what's possible. And sometimes that's perceived as being wacky, but I actually was flattered. I thought it was a great a great way to say it, because sometimes that is how things look when you see it for the first time, because the nature of SBX, and I can talk about in a bit, what it is and describe it in detail. It's been kind of a long journey to get here. And if it's okay, go back a little bit on the history here. My inspiration was PCR at first. Not surprising that SBX ended up at Roche. Right?
Yeah, the PCR champions.
PCR champions. So, it was not a surprise at all, but also manual Sanger sequencing. When I first did manual Sanger sequencing, I was hooked. I thought this was the most amazing thing. This is, 35 years ago. This is a long time ago, but I just thought it was amazing. And it really inspired me to think about, look at how powerful this tool is. Look at what you can do. Tool, and great, you're doing a couple thousand bases in a day at best, but it kind of led my thinking about, and how I was inspired to think about technology and the impact that developing tools can have. And so, I got really lucky in that my first biotech company I worked at, called Darwin Molecular, in the mid-90s, the guy heading that was the guy who started the Human Genome Project at DOE, David Gallus. And so it was just a great place to be. Leroy Hood was one of the scientific founders of the company, and it was just an inspiring place. And in fact, a couple of my coworkers just won the Nobel Prize. 2 weeks ago, or a couple of weeks ago, Fred Ramsdell and Mary Brunkow won the Nobel Prize. It was an inspiring, great place, and I was lucky to be able to be there and have that experience. And in terms of the work I was able to do, also really lucky, because I got to work on protein engineering and developing tools. These are genomics tools. These are all really important things if you're going to think about making a sequencing technology, especially single molecule sequencing technology.
Okay, so you've been there all along with all the major advancements. You worked with the great players.
Yes.
You're just a bit of a late bloomer.
Yeah, I had hair at the time, it was yeah, yeah, long hair actually. It was funny. But yeah, late bloomers. So it's always been there, kind of thinking about, how to solve this problem of sequencing. How do you advance sequencing? And in all of this, I actually it's kind of funny story is I, we spun out a company during that era at the end of the 90s, and a company called Rapogene that sold to Qiagen. Well, the guy who was the president of that little spin out was a guy named Nick McCook, who was the CEO, became the CEO of Solexa as soon as he left from there, and which then became Illumina
Illumina.
technology. So, I talk to Nick every once in a while, and he was an inspiration to me as well. And so it just kind of things lined up that this was going to be something that was always an interest in wanting to do. As I
So,
went through, go ahead.
sorry, I'm curious. So it's always been a project in your
Always.
mind. You've been working away on it.
Yeah.
2007 is when you say you came up with it. So in the meantime, you're looking at all this incredible stuff going on in sequencing, right? It goes to single molecule. Then we find out that PacBio didn't have great accuracy, but if you stacked it up, through consensus, then it had fantastic, and they promoted what they call long reads. And fast forward, then we get Element and Ultima coming on the program. I'm curious, did you think, oh, it's too late? I mean,
There were moments. I mean, I'd always have to, every time a technology would come out and they had raised so much money, I'd have to go to the board and say, okay, this is what we think about what's going on, and where the limits are going to be. And I have to say, we kind of nailed it in most of the cases. We predicted some of the challenges that the other technologies would have. And so yeah, it was always hard because a little bit about this when we went out to do our fundraising, September 2008. So
mm,
this is Stratos Genomics. So a week
yes,
after
that's
Lehman
right.
Brothers collapses, we're going to go and raise money. So again, this set the tone. We had to figure out how to survive and keep advancing the idea while other people had raised money already. PacBio, these others, they'd raised some money, and so they could keep going during that phase. And so we just kind of figured it out, and that led to kind of a certain rigorousness and scrappiness of our approach. And I think that was necessary to keep inventing, because there's a lot of inventions that had to come together to make this work.
Okay, so let's get into it.
Yeah.
Let's talk about what it is. Give us the, clean bench level explanation of sequencing by expansion.
Yeah, it's kind of funny. Our approach to efficiently sequencing DNA is to not sequence DNA. That's the funny part about it.
And
Is
what does that mean?
Yeah, exactly. So, I mean, as you know, DNA is spatially compact. Chemically, the bases aren't that well resolved. So, from a signal to noise to do single molecule measurement, that's really hard. So, my idea was to just rescale the problem, expand the molecule. So, that's why we call these things expanders. So, just expand it by 50x. Okay, so how do you do that? So,
That's kind of the principle of PCR.
in a way, yeah, because whereas PCR is increasing signal to noise by increasing the number of molecules, we're increasing the signal to noise of the measurement, but by expanding that structure. And so this is a biochemical process that uses a thing called expandable nucleotides to just recreate a new surrogate molecule that's expanded, that has these reporter molecules that you can measure in higher signal to noise. We were always thinking about nanopore at the time. We were thinking that that would be what we
You
would aim.
were? Okay. Yeah, because I saw you got bought by Roche in 2020. I know Genia was bought prior to that.
Yeah.
So I'm curious when you start thinking, oh, we got to do this by Nanopore, and maybe Genia's the right partner.
When we started, Genia actually wasn't around, but a nanopore was starting to grow some steam. We thought, okay, this is a good fit, but hey, we could apply it elsewhere too. But it made logical, always made sense that that was probably where we were going to land. So when you make these structures, you engineer them to be compatible with the measurement modality. In this case, we thought nanopore. So, imagining that you have this expanded structure, you have to put in control elements so you can not only just thread the molecule through, because these are 20 megadalton structures,
Mm,
massive molecules that directionally thread. You have to control the measurement very exquisitely with voltage, and then measure them over and over again. So you get more and more molecules going through. That's how we get this throughput. It's a serial measurement where you measure one molecule, then the other across millions of pores that came from the Genia technology. So millions of pores. That's how we do it. Tons of innovation, not just in that initial chemistry, but throughout to pull it all together.
Okay. So the larger molecule gives you more signal to noise.
Exactly.
Now,
And
why
less
hadn't that been
noise,
done
not much
before?
better.
And why didn't other sequencing companies come up, think about
Yeah,
that, or do they need it?
yeah, no, I think it's insanely difficult to do because even just the nucleotide change, you're violating the rules of chemistry and changing the electronics of the nucleotide just to make the structure. And then you have to have an enzyme there because it's an enzymatic replication process that recognizes it. So we had to do a whole range of protein engineering and molecular engineering and bring all that together with new innovations just to actually make that process work. Because it was really challenging. It was not at all obvious that you could ever do this. And that's the thing I like about it is we showed people kind of a next level of what's possible by making the technology work, which is
Uh-huh.
really, yeah.
Uh-huh. Yes, absolutely. That's what it feels like. Now I know Genia was always working on scale. Was scale your big throughput, your big driving vision from way back then?
Scale and flexibility. Flexibility was top of the list. We wanted something that was like, okay, if I want to run a small amount of sequence, I can do it. And it's right at my fingertips. I'm inherently impatient, so I want to be able to get results faster. And I thought that
Dude,
was
you are a bar guy. You make bar
yeah,
bets, you violate all the rules, and you're impatient.
exactly, pretty much. Yeah, no, for sure. And I think, but those are the founding principles. Scale for sure. Flexibility, though, right at the top, because we saw that as where it would evolve eventually.
Well, congrats on partnership with the Broad, which I mentioned, and Boston Children's. It set a Guinness World Record for fastest whole genome. Um, what? So from blood to variants. So what? There's part of that process that you could speed up, I think, to 20 minutes or something, right? And then there's a lot of that process which you have no control over. Is that
Yeah,
right?
yeah, I can give a little more detail. It was fun really to go for this world record. It was a great project. And but I'll be a little more about me. I always wanted to get the world record in the shot put, but it turns out I wasn't very good at it.
In the
So
what?
the shot put in track and field. So
Oh, the shot put.
yeah,
Okay.
but I wasn't very good at it. So 35 years later, okay, here we go with the world record in sequencing.
Yeah.
Um,
If you
yeah,
hang in there long enough, and be open to other ideas, you'll get it.
you can come up. Yeah, exactly. No, you're absolutely right. I mean, there's certain steps in the process. So to do from the sample all the way to sequencing, there's library prep, sample acquisition. We have to actually make this expandable molecule. So those all take time. So the team in Seattle, my team, compressed all of those time windows down. The actual sequencing part of this was like 15 minutes. So it's really, really fast. We put the sample on, the sequence goes. And on the CSI side, the accelerated compute, we're able to do the analysis in real time. And so just very, very fast. It's literally as the sequence is coming off, we're analyzing it, we're doing the alignments. And so it goes into secondary and tertiary very quickly thereafter.
Is
It's
that you part of your technology then? That initial analysis?
Yeah, absolutely. I mean, and that whole pipeline, that accelerated compute is kind of hand in glove with the measurement. So we can do this real-time measurement. You can actually start analyzing the data, doing demultiplexing and alignments and all that, while the data is streaming off the system, which is really, really powerful. And that's how we were able to do this fast.
Okay, yeah, because that takes more time than the sequencing traditionally, right?
Yeah, and then so that acceleration, and again, this confluence of bringing together this GPU and this processing speed, accelerated compute, just the timing was great because now we have this capability and we can get that accelerated compute really coming together. So as soon as the sequencing is done, the data is there, ready to move on to secondary and tertiary analysis, which also is accelerated.
Okay, so looking at your marketing specs, which are pretty stunning, 7 genomes in an hour at 30x. Translate this into practical throughput for like a clinical lab running with mixed sample types. What's the real world genomes per day, cost per answer kind of answer?
Yeah. So if I answer that, I'll get a zip through my computer from the commercial team. But I'll say that the marketing specs and all that, those will
Right,
come
right.
out.
Yeah, it's early days to say that.
Yeah, yeah. But I can say a few things on this. So what we've shown in early access in-house results are amazing. F1 scores, coverage, all of that. And these are things with those 7 plexes, those genome and we do this all the time. And we also do this with other, more samples, customer samples in their labs with early access. And, we're very, happy with what we see with the workflows. And that's one of the points too, is that, the flexibility and the capabilities that we have with SPX, people are going to be able to rethink their workflows, how to better leverage this flexibility, time to results, throughput, and how to distinguish between the modes of sequencing. We've talked a little bit about duplex and simplex sequencing maybe at some point, but how do you do all of that? And so one practical example, just to maybe give you a little bit of bearing on the question, is we've done tumor normal samples sequencing at Hartwick, one of our early access customers. And, they would want to get, say, 90x versus and 30x coverage respectively for tumor normal samples. So if they get 6 samples coming in, they can make a single expandomer, do a 4-hour sequencing run with our accelerated compute, kick out some cram files, and then they're off and running. So you don't have to wait for a couple days to collect more samples. So that's kind of like a small lab example of that flexibility that you could bring. But you can also bring the workhorse, turning out the crank, because this has massive throughput where you're just running 24/7. And so you get both options, I think.
So that's what you mean by flexibility is you don't have to do big scale.
Exactly. You can do what you want without that kind of penalty. And I think that is for smaller labs. That's a huge advantage.
That
And
is cool.
people
Okay, that I
really,
was wondering what you meant by flexibility. Okay,
yeah,
so throughput's a big one in our field. Accuracy is the other thing, and then the third one is the rise of long reads. So let's
yeah.
talk both of those. Accuracy.
Yeah, actually, that's an important question. So, as I mentioned before, SBX is a single molecule sequencing approach. So, what we see with our raw reads for simplex raw reads is about Q20 plus reads. Okay, which is amazing for single molecule, but it's not good enough. So that's where we came up with the duplex sequencing, SBXD, which means
Okay.
that we're actually reading from a fragment of DNA both original parent molecules on the same sequence read, the expandomer sequence read. And this allows us to do intramolecular consensus. And that's how we get these amazing F1 scores and quality scores that we're seeing. So we use the duplex sequencing as our high accuracy. But then simplex is great because it allows us to do shocking throughput for the short reads, like billions of reads in an hour, and longer reads that can get into that 1,000 range. And they're still really, really high throughput. And so these are the kind of things that we're balancing between. But yeah, the accuracy question is it's for single molecule sequencing, I think it's an important one to think about. And but there's as we know, there's trade-offs between that duplex
Yeah.
read, a little bit shorter, so 250 range of the insert size. That's kind of the length. But we get longer reads in there too. But then the simplex, those Q20 type of reads, those can go out to 1,000, 1,500. In fact, I've been presenting data on that at conferences just, at ASHG, just presented a couple data sets for bulk RNA, longer bulk RNA sequencing. So it's kind of like those things coming together. Again, another powerful advantage of the technology is you can push a little bit longer and accurate.
Okay, so with the duplex, and you say that's shorter reads, but you're getting more consensus. Compare that to PacBio, who, I understand on the program, if you're going for accuracy, they're sort of they're a gold standard.
Mhm. Yeah, I mean, the comparisons, I don't know, I can say that, what we've shown in terms of our F1 scores for our duplex sequencing, consensus coverage and all that, I think it's looking, quite good in comparison to what I'm seeing. But I
Okay.
think, yeah, I mean, that's kind of what, from my viewpoint. But it also comes down to, and this is always the case, I think, this is efficiency and cost efficiencies, workflow efficiencies, that all have to come together into the argument, for why you sequence with a certain technology.
That's true, especially at a very high scale, high
Yeah.
throughput. Now, I
Mhm.
remember Oxford Nanopore struggled with accuracy for a bit. They, every year they'd get a little better. And I wondered if that was something to do with the nanopore itself. But you guys are coming out the gate with pretty good accuracy.
Really great accuracy for single molecule. But I would say this is something that we're going to keep improving. We know the buttons to push. I mean, because we've been doing that for many, many years in terms of how you increase the accuracy of that conversion step as well as measurement, that actual sequencing step. So we know those things. We know the things that we need to do to push length further. So there's a lot of that. What's if it ain't broke, don't fix it. Just we're going to keep doing the things that have
that
allowed
got
us.
you here.
Yeah, exactly.
Methylation epigenetics has been one of the big topic on the program this past year.
Yeah.
Innovations in that. PacBio had 5-base sequencing a while ago. Illumina came out with 5-base recently, and we have a program coming out on 6-base sequencing with a company called Biomodal. Will methylation be integrated? What do you think about that? What's your long-term multi-omic vision?
Yeah, no, great question. So, I mean, we're definitely understand that methylation is important and quickly evolving. And we actually recently did a collaboration with Watchmaker using and testing their TAPS Plus chemistry kit that's coming out in the next couple weeks. So we got a kit, we applied it to our SBX duplex workflow, and I'll tell you, we're still smiling at the results because that the TAPS chemistry converts 5-methyl-C and 5H-methyl-C to T. So you don't have you're able to maintain genomic complexity. It's not like you're converting all the bases to T and you're reducing the complexity of the genome. And what this allows us to do is to do concurrent detection of DNA variants and methylation signal from the same library, which is just great. So it's very efficient that we're able to do that. And that read structure allows us to do reference-free methylation detection. And so once we assess that methylation pattern, we can then take the converted bases, revert them for mapping and alignment purposes, and get the variant calling results, all from one test. And so I presented that at ASHG. And I think it got a little bit lost because we did the world record. I think everyone was focusing on the world record part.
Oh, okay. Well, that's why we're having you on the program here.
Exactly. No, it's great because I was really excited to show that data. We were all just giddy, I think was the word I used. So happy, I think with how quickly. I mean, and that says a lot about the technology. Take a kit, we applied it, it worked beautifully. That's a robust technology, and I think that was on both accounts really. So I think more to come on that, and I think, but it's not to say that we're limiting to that. I mean, we're going to look at all chemistries out there, including ones we work internally on, because we know this is an important thing into the future.
This all sounds great. Congratulations, really. It's a major story. When will it be available?
Launching in 26. I can't give specific dates on that, but that's the plan is in 26 to do a research use launch. That will be our rollout for broad commercial rollout, research use.
Research use. Okay, so tell me more about the rollout as much as you can. Like, who do you think will be your first customers? You talk about this flexibility, so it's not just big labs like the Broad.
Yeah.
You're going to talk about the part of the market that you're going toward.
Yeah, I mean, when you ask me, my vision is everywhere.
All of
That's
it.
going to be my thinking because I think it's a universal. That flexibility isn't just limited to one or the other. Everybody kind of, I think, is going to be able to leverage that capability. So I really want to broadly change and enable sequencing technologies into the future, and that's kind of how I look at it. So I'm hoping that it runs the gamut because I mean that's the thing I like about that we're doing research use only is you get it out there and there's going to be things that people are going to learn to do that we hadn't really been thinking about.
Oh yeah, we've watched this with the other technologies. The new instrument drives new science.
Exactly.
Yeah. So I'm curious about long reads. We had Alex Dickinson on the program, and I don't think he has a boss these days, so he says whatever he wants.
Yeah.
So he said he's just a podcaster. So for whatever that's worth, but he said the reads are more medium, the long reads, right? What would you say about long read applications which have been developed, say structural variation, and this kind of thing? Are 1,000-base reads up for that?
Yeah, I mean, I think our plan is to keep pushing out length. I mean, go back a couple years, we weren't at the lengths we are now, and we'll keep advancing and pushing in lengths. But I think that is a good question. I mean, if you get billions of cost-effective reads of a certain length, how much of that puzzle can you fill in versus the longer reads that go out 10,000, 20,000 bases? And I think that's going to be something that we're going to keep working through. I'm really happy that we can offer so much value and be able to offer that value of that high throughput, longer read lengths, longer. That's what we're talking.
Longer, high throughput, longer reads. Yeah.
Yeah, like on the order of billions in very quick amount of time. I think there's a lot of value in that. But I'll tell you, we're going to keep pushing length. And I think there's a lot of things we can do to keep pushing length. We really only got started on this in the last year in earnest on pushing length. So
What do you think will be the near-term killer app in oncology or maybe rare disease, where SBX's speed and accuracy just change a clinical decision in the same day?
yeah, I mean, at least as far as the killer app, it's that's always hard for me to pick. But I mean, I love what we're doing with MRD and fast and a whole range of things. But I mean, I'd kind of say I'm excited to see what comes out of RUO, what comes out of that and the learnings from that. That's why I'm so happy we're doing it that way. And then really leave it to others because like we said before, I think there's going to be new apps that are going to come. As you said, technology kind of drives what people are able to do. And I think that will be one of the things I'm interested to see come out of this. But in terms of performance, the things that I see, I'm really happy with the direction we're going. I think we're going to be able to have maybe lots of killer apps, but we'll see. I think very interested to see what the customers do with it.
The price you've predicted is going to be around 750,000. How does that fit in the market today? I remember back when PacBio was around that for, the early sequel or some of the early floor models. Yeah, where do you think that puts you? Is that the big labs? Does that get you into anywhere? One thing I know about labs who do sequencing, they want every single technology available.
Yeah, that's true. That's true. I mean, I like where we're positioning on this. I think considering the output and the capability of the sequence, I think it's kind of it's a great value point there. But I mean, again, that's a commercial discussion and to kind of talk through that part of it. But no, I think I think that's the reason why I like where we are is that's a massive workhorse sequencing capability on the one hand, that's compete with anything out there. But then you have the flexibility all in one. So you can do the whole range, and with, I like to say, almost with impunity. You can run down that throughput spectrum and flexibility spectrum like we talked about. And I think that's a great advantage for SBX. So I like where we're positioning ourselves.
What data are you seeing in FFPE and small input DNA, such as cell-free DNA?
Yeah, I mean, I presented on last couple conferences, both MRD results, which are great. FFPE did even some comparators on that in terms of our, able to see, structural variants and our Fred scores and the quality of that relative to other comparisons. Really liking it, and it's advancing quickly. We only started doing FFPE in the last few months, and the data I'm seeing is quite good. Quite good. So like where that's going, I think there'll be a lot more in the coming months on that I think will be coming up, whether it's early access presentations by people. We've already had some of those or internal or both data sets, but I'm really liking the direction of that.
So let's say we talk to you again after you launch, in at least a year. What does success look like? What's your goal in a year? It's launching, but flesh that out a bit.
For me, what success looks like, I mean, honestly, I mean, this is coming from me. I think this is, SBX will be becoming the new standard in sequencing. I mean, I'm really excited to see, like I said, what customers do with this technology. That's a whole other layer to it. Like we have our perspective, but these are great scientists that are out there using this stuff. They're going to bring their own take to this and see what they do, see how they're implementing at scale. They'll probably do a better job at implementing at scale. They think about it than we could. See where that goes. Test new applications. Come up, do things that again, like we said, we hadn't even thought of, or maybe couldn't be enabled with the existing technologies. And to me, I think that's where the success is. I mean, because look, I mean, innovation can't stop. Yeah, I think we know this. It needs to keep going. It will keep going. And as we said, you keep improving the tools, people will figure out how to use them and leverage that. And because at the end, we have a lot more to do. Biology is complex, and I don't think it's going to stop at where we currently are. Like for me, I talk about trillion read experiments. I'm serious about it. I think there's value, I think, in being able to to push that envelope. So as we have to keep evolving, keep innovating. So
I love that point about, the story still being written, because the big second part of the story is customers getting this in their hands and having a lot of fun, giving
sure.
you feedback. I've hung out at Illumina, I've hung out at PacBio. They both feel like scientific institutes. The collaborating with scientists, is phenomenal, because, like you say, you get a Chris Mason on this, or one of those great scientists, Mike Snyder, and they're setting this up in their own way, and they've been doing that longer than you.
Yeah, for sure.
Yeah,
No,
that's a great point. What's your biggest challenge, going into launch?
yeah, I mean it's like anything else. There's a lot of pieces that come together for any technology at launch, and so there's it's all the same things that anyone else would have. But I mean, I think we're well into it. Got a great team of people pulling this together across the organization. A very experienced organization at launching products. So I think it's but it's like anything else. Sequencing technologies are complex. There's a lot of pieces that go with them. So making sure you pull all that together. Those are the same challenges, I think, as anyone would have.
Roche, speaking of the company, Roche usually takes a pretty long view
Mhm.
with regulatory items. Any thoughts on that at this point? I know it's early days.
Yeah, I mean, look, when Roche acquired Stratos, we were talking about IBD.
Yeah.
Uh, but I mean, as we said, we're launching with RUO, and I'm really glad we're doing that for all the reasons we discussed. But I mean, a lot down the road, of course. I mean, Roche will pursue IBD and go down that road. But where we are now, I think I'm really glad we're going down this track, because I think there's a lot of value and a lot of enablement that can happen with getting it out there in that format.
So I'm curious, is there any question I haven't asked? Any part of this that
Oh,
the you're dying to tell me that we haven't talked about?
yeah, I mean, I just think, as a kind of go back to the beginning part of this about how these things kind of come together. I mean, it's unlikely that we're here. And I mean, there was a certain, I don't know, what was that? I don't know if you've ever watched the Avengers movie, where at the end there's like only one way that they can come back and return things back out of the 14 million combination. It feels like we with SBX we kind of had to go down a certain path to get here. And it was really an unlikely way that we went from Stratus Genomics and my co-founders Al and Bob, and to getting to this point. And I think it just says that there's not just one fixed way of innovating and making new technologies. We kind of did it a different way, very different, and broke a lot of rules, as he said, in doing this. And, it's kind of a fun, exciting story to tell, and I'm just glad that we got here, got to this point, and are able to see very soon people getting it in their hands. And to me, as a sequencing technologist, going back to the original dream, that was everything. Just seeing people look at SBX in awe and being inspired to do something even more next level with their work. To me, that's everything. And that's the message for me is, and also the hidden message of, SBX works. Now what else can people do? Be inspired to do? What else is even the next crazy idea? So
Well, let's finish up with that. Talk to me a little bit about that next world. I remember when the CEO of Ultima, Gilad, came on a couple years back when they came out of stealth. Surprised everybody.
yeah.
Um, and he was all about scale. He'd come over from tech, and he had seen that scale made so many things possible in tech, right? Just that Moore's law and just got to scale. And this is always what we've said about sequencing too. It's going to enable so much more in the clinic. Um, but I remember asking him, is the problem with genomic medicine really scale? Is it that so you talk about trillions of bases.
Mhm.
Um, is that really the bottleneck? I'm curious your thoughts on that.
I mean, I think you have to be able to, there's certain questions in biology that you really do need a certain scale. And I think, as we said before, once you enable a certain capability, people will say, oh, well, now I can do these kind of experiments and I can do it at a reasonable cost. And so there is always that part of it about the overall, science, driving science part of that equation. And I think that's certainly one part of it. But that's why I always emphasize flexibility too, and the ability to do and get and answer questions quickly. Because I do think a lot gets lost, I think, as you're doing either research or even on the clinical side eventually down the road, by the timing factors of being able to get results, timely results. And I think that's why I've always, again, it was a founding principle of SBX was flexibility, being able to sequence differently, than other technologies that relied on sequencing by synthesis, for example, where you have to sequence over a certain window of time. And so I think that's certainly a big part of the future and the new future of sequencing is being able to operate with that flexibility, but also at scale, at being able to do really high scale, high throughput. And I do think we will keep pushing the boundaries there and advancing. When you think about perturb-seq type of the scale and the massive amounts of sequencing to do there, you need something that can do that. But then you also want to have a technology that can do those 4-hour genomes or even less, something even faster than that. So,
Mark Kokoris, head of the SBX program at Roche. Hey, thanks for taking some time for us.
Thank you so much, Dan.